Hello, welcome to Statistical, and today we're going to be talking about how do you pick up marks in biology. Guys, it's an absolute nightmare, yeah? You've learned the topic, you know what the topic is, you write it in the exam and you're seeing 2 out of 6, you're seeing 1 out of 6, and you're like, what is going on here? So, today I'm hoping to go through all the different types of questions that you'd expect in a biology paper, break it down so that you know how do I answer, what is the examiner really looking for me to do in this question. So, let's try to get into it right now. Okay, so I think the first bit to mention is going to be about how we are assessed in biology, right? So, SNAB, we've got 3 papers. We've got paper 1, topics 1 to 4, 5 and 6. We've got paper 2, which is topics 1 to 4, 7 and 8. And then we've got paper 3, which is all 8 topics, the big one. Now, you should know which topics are in each paper, but what people kind of miss in that really long exam spec, which I'm sure everyone's read, yeah? We've got the assessment objectives. So this will be your AO1, AO2, AO3. We'll get them up on the screen as well. But AO1 is going to be more just like, it's just knowledge that you could have just by revising the topic, yeah? AO2 is going to be more about taking that knowledge and applying it, whilst AO3 is going to be about analysis questions. It's going to be like your graphs and calculations and stuff like that. Now, with what is going to come up on your paper, at some point, all of them, yeah? But AO2 tends to be the one that's assessed the most. AO1, AO3 is kind of a bit more similar in terms of how frequently they're assessed, but your AO3 is going to be much higher marks compared to AO1. So I should be able to get that up on the screen. Now, there are a couple different types of questions that we have in biology. I should be able to get a list. If my editing's good, I'll get a list of all of them to the side here, and we're gonna go through them one by one. So there will be timestamps in the video. If you feel like, okay, recall, I'm quite confident on that, but data analysis, I need some help in that, you are able to skip to different parts of the video to the questions that you think will be most useful for you. So let's head over to the whiteboard now so we can explain a bit more about what is going on in these questions. Okay, so our first type of question is going to be recall. Now guys, these are the easiest questions that you're going to get on the paper, yeah? They're literally just going to ask you to name something that you should already know. If you've covered the content, you should be able to whiz through these questions. Shouldn't even take a minute. So for example, all the answers are C. As you saw at the top of the paper, we're only being assessed on AO1. So guys, do not stress about these questions. If you've done content, you should be able to get them. And to be honest, in terms of the grades that you're trying to get, guys, these are not questions you want to drop marks on. So make sure that you learn your content well so that you can just breeze through questions like this. Let's move on to the next type of question. Okay, let's talk about the describe question. Guys, again, this is one that you really should just be picking up the marks on. It's not too difficult. We've got here, describe the structure of a DNA mononucleotide. This should just roll off the head. Deoxyribose, nitrogenous base, phosphate group. Only thing that I'd maybe add is just to add that it's adenine. A describe question is literally just telling you to explain something, yeah? If you've covered the content, you should be able to cycle through that quite easily. And just here, we've just got another recall question. Not too much of a big deal. Again, if you've learned the content, you should be able to answer that quite quickly. Cool. Okay, so the next type of question that we need to address is your explain or your suggest questions. Now, whenever you have explain or suggest, what you're kind of thinking is, okay, I need to make a point. So we'll highlight this. I need to make a point and then I need to justify it. So you make your point and then you have to justify it. How have we done that in this example? Point in blue. So we're explaining the importance of dipole nature in sweating. Dipole nature, what kind of is that? Oh, why is it relevant? You're allowed to make hydrogen bonds between water molecules. That's our point. Why is formation of hydrogen bonds relevant to sweating? Yeah. Whenever we sweat, we want to release heat. We want to cool down. So the energy needed to break the bonds, the hydrogen bonds between the water molecules, allows for evaporation. And when water evaporates, you release that energy, allowing for cooling. So do you see how we made the point about the dipole nature forming hydrogen bonds, and then we've justified why it's relevant to sweating? Because obviously the aim of sweating is to cool down. Okay, so now we're going to get into our data interpretation. Guys, this acronym is a beast. Chart C. Okay, I want you to focus on this. So, whenever we have a data question, you're going to want to compare, look at the differences or similarities between the groups. You're going to find your highest or your lowest values, yeah? The maximum and minimums. Anomalies, any result that's unexpected. Rates of change, how quickly or slowly things change. The trend, overall patterns, and do a calculation. Easy as that. So remember your chart C. if we go here to the question, I've already drawn on these lines of best fit just to make it a bit easier for myself, but they weren't there before. Let's look at it. Let's do some comparisons, and then we'll relate back to what the mark scheme said. So if we do some comparisons, I can see, if we just focus on, if I just get my highlight out, let's focus on the people that are exercising first. Cool. So we're looking at the circles and the triangles. I can see they're all above the X's or the people resting at every single temperature. So I can say that the rate of sweating, remember, relate it back to the X and the Y, the rate of sweating is always greater for people exercising compared to those at rest. Cool. That's one thing we can say. What other comparison can I make? Well, you can see here, if we look between the circles and the triangles, they're all quite close to each other. And you can see that by the lines of best fit too. They're quite close to each other. So I would probably also mention something like, there's not much difference between exercising and exercising twice as vigorously, for example. Cool. In terms of comparisons, that kind of seems to be it. I mean, you could also maybe say, yeah, let's leave it at that for comparisons. Let's look at highest or lowest. Now, you need to make your own judgment. Is it relevant for this question or not? In terms of highest, you can say that I don't think it's too relevant for this question anyway, but you could mention the highest rate of sweating occurs at the highest temperature. You can maybe mention that. I don't think it's too relevant for this question. Now, anomalies. Let's look at this. Cool. Anomalies, as you can see on our graph, I wouldn't say that there really are any anomalies. I mean, these 2 points here, maybe you could try to make an argument. I feel like it would be kind of weak because you would expect less sweating to occur at lower temperature. So I don't necessarily think there's any anomalies here. If we saw an X up here, then maybe we're thinking, okay, that's bizarre. We need to mention that when we're answering the question. But I wouldn't necessarily say there's an anomaly on this chart. Let's look at the rate. Now, in terms of rate, I can see these lines that we've done, the lines of best fit for the exercising and twice as vigorously as well, they're just going straight, that's linear. Whereas this one for resting, you can see it's quite low. It's quite low to begin with, and then it starts to pick up afterwards. I'm mentioning that in my answer. That's definitely something that I've seen. Yeah, I can see here that the rate of sweating starts to increase by more, or there's not much change until you get to higher temperatures at rest. Yeah, so the rate of sweating doesn't increase as much until you get to higher temperatures at rest. That also kind of links onto trend. Trend, I just want to see the overall pattern. You can see in all of these, at higher temperature, the rate of sweating increases. I don't know if we're going to get a mark from that. I definitely mention it. Last thing to do is a calculation. Guys, if you get marks for it, sometimes you will, sometimes you won't. Just mention it, okay? Just do it. Cool. It could be finding the change in the rate of sweating over some temperature. So we could do a rate calculation. We could just find the difference in sweating between two temperatures. Anything like that could potentially score you a mark. So I just say do it. When we look back to the answers that were on the mark scheme, rate of sweating increases with higher temperature. Guys, what does that sound like to me? If we get to our purple pen again. To me, that sounds like we did a comparison. Or we did a trend. Because the rate of sweating increases with higher temperature. We mentioned that as a trend. At any temperature, sweating is greater with exercise. You could say that's comparison. It's comparing it to at rest. Cool. Only a small difference between exercise and exercising more vigorously. That's another comparison that we mentioned. And the increase in sweating begins at a lower temperature during exercise. Now, this one, you could either say it as a trend or you could say it as a rate. I would maybe lean more towards it being a trend. Yeah, you saw that the graph wasn't linear compared to the other ones. Guys, that is kind of how you want to approach a data interpretation question. I hope that makes sense. Again, any questions, just leave them in the comments. Let's move on to the next type of question. Okay, so we're going to deal with a calculation question now. This one, you're doing a statistical test, chi-squared. Look, they'll always give you the formula, so you just need to put it into the actual equation. Yeah, you just need to do it properly. Cool. If we had the previous part of the questions, you would get the context, but we're going to look at the expected number, the observed number, and we're going to put that in. So we're going to look at this top row here. That's just the context of the question. That would have helped you to get it. That's that one. Cool. You put in the values just as they are, put them into the equation, you get your answer. I would say rounding to about 3 significant figures is always a good idea when it comes to this question. But the main reason I wanted to introduce this is because we need to focus on this. Now, this comes to the data interpretation part of it. Guys, this is something that you need to know, statistical tests. It's going to be the number of groups. So you can see there was 1, 2, 3, 4 groups. -1, that gives you your degrees of freedom. So we're focusing on 3 degrees of freedom, and always, always, always use a 0.05 as the percentage, unless you are told otherwise. So our critical value is going to be this. Now, here's why we need to know the context. We need to know, okay, our critical value that we had in the last question is less than the critical value here. So the chi-squared value is less than the critical value, meaning that there's not a significant difference between observed and expected results. That's just something that you need to know in all the cases. If our value is lower than the critical value, there is not a significant difference. Cool. And then the second part, context of the question. So this part, again, we go back to the concept of the question. Traits are inherited independently. I haven't put in the stem of the question, but that's how you would know. I hope that makes sense for answering a question on a statistical test. Okay, so when it comes to practicals, we need to be very specific about how we answer these questions. But if you follow this kind of structure, you probably will be scoring up to 5-6 marks every single time. So what I like to do, to be honest, this was the paper I did when I was doing my exams. I thought this was such a mean question because technically you don't actually do this experiment. They should ask you questions on experiments you've done. Nevertheless, let's go and answer it. So I always start with the variables, dependent what you measure. If you looked at the context of the question, it would be the time from hatching to pupating. Yeah. Independent variable, what are we changing? The temperature, because we're studying the effect of temperature. If we highlight it, we're studying the effect of temperature on the duration of the larval stage, right? So we need to change the temperature. Now, guys, whenever you have something like this, you need to be so specific to get your marks, yeah? State the range of temperatures you're going to use. So like, for example, 5 degrees to 40 degrees. In fact, I'd even go one foot further. I would say we're going from 5 degrees to 40 degrees Celsius, and we're going in increments of 5 degrees, for example. Cool. Control variable, just mention 1 or 2. Just mention 1 or 2 control variables. We'll get you some marks. Cool. Let's go on. This one I like to add. You don't necessarily need to add equipment. I think this is a nice chance to just put in some additional points maybe. So like, for example, when you have your root tip squash practical, it would be mentioning that you use hot hydrochloric acid. Something like that just picks you up a few extra marks. It's really good to mention. Now the method. We aren't going to go into the method in this explanation because it's not too relevant. But what I really want to emphasize are these points here, 5, 6, and 7. Guys, please, please, please always mention these. So you'll do your method as normal. If it's a practical you've done already in class, just go through what you did. Yeah. I want you to mention repeats. So crucial. Get to your mark every time. Just mention that you do repeats. Calculate the mean. Do a statistical test. Just add them in. Even if you didn't do it on the day of the practical, add it in. It will get you some more marks. Yeah. That's kind of how I want, or how I would structure answering a practical question to get your marks. Cool. Okay, so now we are on to our evaluate questions. I believe these are some of the hardest to answer when it comes to our A-level. So we're going to go back to the sum of the question. Or let's read the question first. Evaluate the information provided in the table and graphs to determine which factors are most likely to increase the risk of CHD in women. Now, whenever we have anything like this, guys, I start with, let me look at the actual method used to collect data, or let me look at the sum of the question so I can figure things out from that to put my answer. I use CLAP. I don't know why I clapped, but let's move on. Cool. So it's an acronym. Collection method, length of time, age, population size, or characteristics. Let's comb through the text to find those. So our collection method, they can say here, the woman provided information on the following. Yeah. That tells me that they kind of did like a survey questionnaire kind of thing. That's something in the back of my head. It's a survey questionnaire. The length of time, let's see here. They said that the women were monitored for 5 years and the development of CHD was recorded. So I know that it's 5 years. That's the length of time. Let's think about age. We have this in the table here. Our age range is all of that. So 55 to 74. We're going to store that in the back of our heads as well. That's going to become relevant a bit later. Let's look as well for the population size or characteristics. Well, again, coming through the text, I can see, let's highlight it. They selected 1 million women who had not had CHD before the study started. So we're looking at 1 million women, and the characteristic is that they do not have CHD. Those are all things that we've combed through this text and we can put into our answer. Yeah? Really, really important that we do that. Cool. Other things that we want to do. We want to now talk about the data. So we talked about the design of the study. Let's talk about the data. Now, if we look at the data here, we can see
as you're increasing in age, the risk is higher. So at HBMI level, you can see that increasing in age is a risk factor for CHD. Store that in the back of our head. That's what we got from the table. What we also got, increasing BMI is also another risk. Yeah, we've got that too from the table. Cool. Let's keep it pushing. Let's look at our graphs. Now, remember chart C. So important that we remember about chart C. So we're comparing. Now, one comparison we can make here, percentage over 20 years. Well, let's look at the table. The table is looking at over 5 years. That's another thing that we can mention in our answer. We can try to do that about the reliability, the validity of the data. Yeah. Again, what can I see from these graphs? Well, something that we got on the other one that we don't have here, again, one compare, right? Something that we don't have here is the age of the woman. We got no clue how old these women are in these graphs. So that's another thing to mention. Cool. So there we've got some comparisons. We talked about reliability, validity. I'd say get one of them into your question when you're talking about evaluate. So let's just write it on here for the visual learners. We're talking about reliability. We're talking about validity. Okay. What else can we mention? Well, let's look at what's happening in the graph here. Highest. Well, you can clearly see in terms of the percentage incidence of CHD, given that all these graphs are like they're using the same scale, we can see that smoking, such a high risk. I've stored that in. What other things are increasing the risk? Well, they've said that physical inactivity, you can see compared to active, increases the risk. And also, guys, wouldn't expect this. Non-drink of alcohol. Don't go by what you think it should be. Go by what the question tells you. The question is telling you that not drinking alcohol increases the risk of CHD. And that is across every single BMI level. Yeah? Across all BMI levels, the graphs are always higher. Again, remember, compare. We're looking at the graphs. Each graph, the dotted line is always being higher than the solid line. So we can also mention that. There's no anomalies on this graph. Cool. The rate of change, well, you can see all of them are going up. So we can mention that. Trends, we can see clearly that BMI increasing is increasing the incidence of CHD in agreement with the table. Cool. I would also maybe throw in a calculation. I think a calculation from this graph would be relatively easy to do. Just like, for example, you could look at current smoking at 37.5 and never smoking and just do a calculation of the difference between that. That's easy enough for the mark. Cool. Finally, what do we need to do? We need to talk about what the question asked. The question asked you to talk about factors that are most likely to increase the risk of CHD. So if we look at the table is showing us that age. The table is showing us that age has a more significant impact on CHD compared to BMI. And you can kind of see that, like, if we focus within each row, the increase isn't as much as if we go between the columns. Yeah. I hope that made sense. But like comparing 22.5 to 27.5, for example, the jump isn't as big as going from 55 to 59 to 64 to 60 to 64 in the same BMI. Yeah. So you can see age is a bigger risk factor there. Whereas if we look at the graphs, smoking quite clearly has the most difference between the groups from your graphs. So just mention stuff like that. And you can see all of that's kind of backed up by the mark scheme. We talked about study design. We talked about data. Then we related it back to the question. We talked about the risk of CHD. So whenever you're getting an evaluation question, I really hope this made sense. Those are the kind of things that you want to do to maximize your chance of getting the marks. Also, guys, one more thing to mention. They love a conclusion. They absolutely love a conclusion. I'd maybe put a detailed conclusion at the end of it that will get you more into that level 3 to score 5 or 6 marks on the question. So I hope that's helped you with the evaluation question. I know what you might be thinking at this point. Where's the 9 marker? I think the 9 marker is so complex to a point where we need a separate video on that. So this evaluation question is the last one that we're going to discuss for today's lesson.
Okay, so I hope that we've left that video having a better understanding of what the examiners want me to do. How do I approach these questions when it comes to A-level biology? Please like the video if you found it useful. Any more questions, just feel free to ask them in the comments. I'm going to do my best to go and answer them. We've got the Instagram, the TikTok, both at demola. Give them a follow, please. And I'll catch you in the next one. Goodbye for now.